Your brain is made up of billions of cells all working together to help function. However, in order for these cells to work together, they obviously need some form of communication. Just like how we will send texts or letters in order to communicate between each other, your cells use molecules called neurotransmitters, things like dopamine or serotonin, in order to communicate. And this communication is absolutely vital for a healthy brain to function. In fact, many neurodegenerative diseases such as Parkinson's or possibly even Alzheimer's have been linked to a breakdown in this cell-to-cell communication. Max Rice, a current graduate student at Columbia University, is investigating a new form in how cells will communicate with each other.
So my first interest in science really began in preschool, where I attended a program through the Museum of the Hudson Highlands in upstate New York. And instead of nap time, we would actually go on walks throughout the forest and learn about the plants and animals native to the Hudson Valley. And so that kind of started a love for biology that carried on throughout high school. And in 11th grade, I attended a summer camp called Camp Neuro over at NYU, where I learned all about the field of neuroscience and decided to study that in college. And it wasn't until I actually got my first research experience in college that I decided I wanted to pursue a full career in neuroscience research.
When I first started graduate school, I joined the labs of both Dr. Franck Polleux and Dr. Kapil Ramachandran. And both labs are interested in studying the nervous system at the cellular level. And so our body's filled with all sorts of different types of cells, such as skin cells and muscle cells. But one thing that makes neurons, or cells in the brain, special is that they have specialized molecules called neurotransmitters that are released when a neuron is active and can propagate the signal throughout the brain.
In 2017, a researcher here at Columbia named Kapil Ramachandran released a paper called A Mammalian Nervous System Specific Plasma Membrane Proteasome Complex that Modulates Neuronal Function. And so this is really just a fancy way of saying that we found a new structure called the neuroprotesome, which is actually embedded into the outer surface of the neuronal membrane. And it releases a whole new type of neurotransmitter that we have no idea what its function is. And more specifically, this neurotransmitter is formed by the breakup of proteins by this new structure called the neuroproteasome. So the initial discovery of these neuroproteasomes and their ability to produce these new types of neurotransmitters, all of this work was done in cells in a dish, or what we scientists like to refer to as in vitro. And so my research really aims to find these neuroproteasomes and figure out what they're doing in an actual living animal, or what we scientists call in vivo. We're actually investigating the function of these neuroproteasomes in vivo using a technique called GCamp imaging. So GCamp imaging is a technique that involves the insertion of a protein called GCamp into neurons. When the neuron is activated, GCamp produces light, which we can take images of to see which neurons are firing and when during different conditions. And so proteasomes exist in all your cells, and neuroproteasomes are only a small subset, so targeting them specifically has been a challenge for us in the past. Now is an especially exciting time to study neuroproteasomes because we now have drugs that can specifically target them. And now that we have this drug, we have some exciting new preliminary data that shows that using this drug on neurons will actually cause the neurons to fire very rapidly and at abnormal rates. So this suggests that there's some role of the neuroproteasome in suppressing overactivity of these neurons, which is what I want to investigate in a living brain or in vivo. There's also an interesting connection between neuroproteasomes and Alzheimer's disease. The Ramachandran lab has actually shown in their recent work that neuroproteasome dysfunction can actually lead to clumping up of misfolded proteins, one of which is called tau. And this can actually lead to degeneration, and it's one of the hallmark characteristics of Alzheimer's disease. And so getting a better understanding of how these proteins will start to clump up in a neuroproteasome specific manner can help lend interesting insights into the future study of Alzheimer's disease. So my work aims to understand more about how neuroproteasomes function in the brain, which can down the line lead to new insights into future therapies for Alzheimer's disease.
Thank you for watching this edition of Late Night Science. Stay tuned for our in-person events and please watch our other videos. And don't forget to like and subscribe.